In DNA replication, helicase unwinds the parental double helix. Molecules of single-strand binding proteins stabilize the unwound template strands. The leading strand is synthesized continuously in the 5' to 3' direction by DNA polymerase III. Primase begins synthesis of the RNA primer for the 5th Okazaki fragment. Then, DNA polymerase III completes synthesis of fragment 4. When it reaches the RNA primer on fragment 3, it will detach and begin adding DNA nucleotides to the 3' end of the fragment 5 primer in the replication fork. DNA polymerase I removes the primer from the 5' end of fragment 2, replacing it with DNA nucleotides, added one by one to the 3' end of the fragment 3. Finally, DNA ligase joins the Okazaki fragments of the lagging strand. For the initiation of transcription, a eukaryotic promoter commonly includes a TATA box. Several transcription factors, one recognizing the TATA box, must bind to the DNA before RNA polymerase II can bind in the correct position and orientation. Additional transcription factors bind to the DNA along with the polymerase II, forming the transcription initiation complex. RNA polymerase II then unwinds the DNA double helix, and RNA synthesis begins at the start point on the template strand. As RNA polymerase moves along the DNA, it untwists the double helix. The enzyme adds nucleotides to the 3' end of the growing RNA molecule as it continues along the double helix. In the wake of this advancing wave of RNA synthesis, the new RNA molecule peels away from its DNA template and the DNA double helix reforms. At the end, you have a completed RNA transcript. In eukaryotes, RNA polymerase II transcribes a polyadenylation signal sequence which ends transcription.
In RNA editing, enzymes in a eukaryotic nucleus modify pre-mRNA in specific ways before the genetic material is dispatched to the cytoplasm. Both ends of the primary transcript are altered. The 5' end, which is synthesized first, receives a 5' cap, a modified form of guanine nucleotide. At the 3' end, an enzyme then adds 50 to at least 250 adenine after the polyadenylation signal. This forms a poly-A tail. In RNA splicing, enzymes called spliceosomes binds on the introns, removing them from the pre-mRNA. The introns are then degraded, leaving only the exons. The leftover exons are now mRNA. The exons change, and as the ribosome reads the mRNA, a different order of protein is formed, creating a different protein.
This is the first stage, initiation. First, the smaller subunit of rRNA will attach to the mRNA and will read the codon until the start codon is reached. In most cases, the start codon is AUG, methionine. tRNA hydrogen bonds to the start codon AUG. Then the larger subunit will attach. This is elongation in translation. When an amino acid tRNA bound to elongation factor Tu enters the free A site on the ribosome, a trial and error process occurs until the correct corresponding tRNA is found. Elongation factor Tu hydrolyzes its bound GDP, creating energy and forming GDP plus Pi. This then dissociates from the tRNA. The tRNA to give its amino acid to the next tRNA in the sequence. In translocation, the rRNA will move the mRNA to its next codon to continue the process. This will result in an amino acid chain. The ribosome catalyzes the formation of the new peptide bond and undergoes a dramatic conformation change. The peptide bond is passed to the next tRNA in the A site, which immediately after moves to a P site. This requires another GTP. The elongation factor G binds to the ribosome and hydrolyzes GTP by elongation factor G. This will switch the ribosome into this state so that it is able to accept the next tRNA. Once the stop codon is read, a release factor will come in and add a water molecule in the A site to the polypeptide chain. When this occurs, the polypeptide chain will dissociate through the P channel, P tunnel, and with the addition of 2-GTP, everything will break apart.
Gene expression, or the process by which genetic information forms gene products such as proteins or RNA, is regulated by operons. Operator is a segment of DNA that is positioned within the promoter, and it controls access of RNA polymerase to the genes. So in this example, lactose Z, the production of lactose Z is inhibited initially by a protein that represses it. So the protein is attached to the operator and prevents the RNA polymerase from attaching to the promoter. However, when the inducer, or allolactose in this example, is attached to the repressor, it makes the repressor inactive, causing it not to be able to attach to the operator and allowing RNA polymerase to attach normally. So after the RNA polymerase is allowed to attach, the rest of the cycle can continue, and the products include beta-galactosidase and two accompanying enzymes. An enzyme called CAP, or catabolite activator protein, is going to be activated by CAMP, and when it is activated, it will attach to the promoter, allowing the RNA polymerase to attach more easily and products to be made. However, when they don't need to create any more lactose enzymes, or when glucose builds up again in the E. coli, CAMP will dissociate from the CAP binding site, and RNA polymerase will be unable to attach to the gene, causing less products to be made. However, the repressor, if the repressor is attached, then it won't be able to create the product. If it's not attached, then it will be, but if CAMP isn't attached to CAP by the promoter, then it's going to happen a lot slower, and RNA polymerase won't be able to bond as easily.